Hi, my name is Julia Parsley. I'm a Ravi Scholar in Columbia College, and my major is biochemistry. So my research. Right now, one really big area of focus in molecular biology and developmental biology is the mechanisms of stem cell differentiation. While there's been much progress into that field, we do not entirely understand how it is that stem cells differentiate into all of the cells of the human body. Interestingly, however, there is a strong correlation between differentiation of stem cells and changes in this process called alternative polyadenylation. So let me go into that for a bit. There are two main ways in which RNAs are processed before leaving the nucleus, mRNAs. One of them is splicing. RNAs can go through something called alternative splicing. That basically means there are these sequences in the RNA that have to be cut out in order for the message to be continuous. Sometimes you can also cut out parts of the message selectively. That's alternative splicing. Thus, you can make multiple different proteins from the same gene by this alternative splicing method. And the other way in which mRNAs are processed, or the other main way, is alternative polyadenylation. And cleavage of polyadenylation is the process by which an RNA is terminated. The transcription process as it's coming out of the polymerase. It's the process by which it ends. And basically it entails clipping the RNA as it's coming out of the polymerase and then putting on this really long adenine tail. It's called the poly-A tail. Interestingly, it was found over the past couple decades that you can cut some RNAs in different places. They have multiple sites for cleavage of polyadenylation, which isn't something that was obvious. And much more interestingly, it seems that there is a global lengthening. So there are some upstream sites, there's some downstream sites for cleavage. Upon stem cell differentiation, a lot of the cleavage shifts to those downstream sites, leading to longer RNA molecules. So I noticed this and I decided to ask the question of how is that happening? Because it seems to be really important for stemness of cells, this cleavage and polyadenylation profile. Not only is it associated with differentiation of stem cells, but it's also the reverse happens when you de-differentiate cells into induced pluripotent stem cells. Their RNAs get shorter. It's called 3' UTR is the end of an RNA. Their 3' UTRs get much shorter. So it seems to be intertwined with the potency of cells. So my question was, this seems to be an important process, so how is it happening? And I thought that it might have something to do with the components of the protein complex that carries out the cleavage and polyadenylation reaction. We know that different concentrations of those factors in that complex can influence its behavior. However, no one's really looked into the possibility of there being different versions of the factors involved in that complex, different splice isoforms. So I looked into the alternative splicing of polyadenylation factors and looked to see if there were differences in the favored isoforms from stem cells to fully differentiated tissues. So the methods I used for this were computational methods. I was analyzing RNA-seq data. RNA-seq is a way of measuring the total amounts and types of RNAs inside of a cell, so it's a really good way to look for changes in alternative splicing because you can see how many of each splice isoform there are. So I collected some RNA-seq data from the ENA and ENCODE databases, and I put it through a pipeline for differential transcript usage analysis. Differential transcript usage is a measurement of the fractional amount of each transcript coming from a gene. So if you want to find out if there's a change in favored transcripts, you look at DTU. So I came up with a DTU pipeline that I kind of adopted from this paper that was published in 2018. It was kind of a methods paper explaining good ways to do DTU. I used DRIMseq as the final step in the program that actually did the DTU analysis, and what I found was that there are two main polyadenylation factors that exhibit really interesting changes during stem cell differentiation, and they are different stages of differentiation deal with the different factors. So early differentiation, there is a really big splicing change in this one component called CPSF4. CPSF4, also known as CPSF30, binds directly to the polyadenylation signal. It's this nucleotide sequence that is like a little flag to the cell to cut the RNA there. So, when there's multiple polyadenylation sites, there are multiple polyadenylation signals. So CPSF4 you'd think would be very important in determining where to cut. So the fact that there is a really big shift in the isoforms upon early stage stem cell differentiation suggests that might be responsible for the early lengthening of 3' UTRs. And then in late stage differentiation, I found that there is another polyadenylation factor called FIP1. It's actually called FIP1L1 in humans. And interestingly, it exhibits another splicing change, like CPSF4. It exhibits its own unique splicing change. This one specifically, there's a really big increased usage of these isoforms that have a different C terminus. They have a frame-shifted C terminus. That means that at the end of the RNA, the message is shifted in such a way that the end of the protein made from that RNA is completely different from what it is typically. And this frame shift eliminates a region of FIP1 that is the hub for interaction of FIP1 with other proteins. And one really important job of FIP1 is to hold together components of the cleavage and polyadenylation complex. So if you eliminate this region, that interaction, those interactions are gone. So it is possible that through elimination of this region, if the cleavage and polyadenylation complex is destabilized and is less good at picking up PASs, thus increased lengthening of the 3' UTRs, thus the APA profile we see upon stem cell differentiation. So that was my work. That's a little more complicated than that. In the late stages, not only is FIP1 is this frame shifted isoform used more frequently, but there are some kind of depending on the tissue, the CPSF4 isoforms sometimes oppose the FIP1 imposed lengthening, leading to what I think are those nuanced differences in 3' UTR length across tissues. So there's really it seems like there's this like mix and match model going on, whereby you can combine different types of isoforms of FIP1, CPSF4, and then the complex. You can really precisely tune the activity of the complex and how good it is at recognizing polyadenylation signals, and thus you can really fine tune the UTR length. So that was what I found, and it's significant because it suggests that there is this mechanism for it suggests an interesting mechanism for global changes in alternative polyadenylation, which we did not know about before. So that's it can be pretty useful just mechanistically as an educational thing. It also suggests that there might be a role for aberrant polyadenylation factor splicing in cancers, because 3' UTRs are quite short in cancers. They look similar to the 3' UTRs in stem cells. Stem cells also will divide never-endingly. So it suggests that perhaps in some cancers, part of what's responsible for that proliferation behavior is issues with alternative polyadenylation, and that perhaps targeting that could help kill cancer cells. This is all a hypothesis. I have not proved that or anything, but or even have any data on it. But it's just these are the interesting questions that arise from this discovery. But yes, it helps us understand stem cell differentiation. It suggests a possible role for cleavage and polyadenylation in cancer, and it also is an interesting insight into how alternative polyadenylation can be regulated, because we don't know too much about that. So that is everything. I hope you enjoy my poster. I wish I could be there in person to explain it. Thank you very much.